There we are. So the meeting's being recorded now. Yeah, so Miriam did her undergrad in Germany before coming to Dundee to do her PhD with Claire and Robbie on 1314 beta-glucan. And afterwards, she decided to focus more on bioinformatics and started a postdoc position with Robbie on the ERC meiosis shuffle grant, which she's worked with for 4 years. And since last April, Miriam has been working on the BARn project, and that's what she's going to talk to us about today. And I'm sure we're all really looking forward to it. So without any further ado, Miriam, I'll hand it over to you.
Thank you so much, and thanks for the introduction. And let me see. Okay. Happy to talk about the BARn project today. So the BARn project stands for BARley Yield Association Network. And this has been funded as part of an ERA Cups project where Robbie, Niels Stein, and Gary Mühlbauer got together and decided this is something we should work on. And is also funded by each of the individual national funding bodies. And next to me, there are two further postdocs on this, so Ronja and Alison. But as you can see, there are a lot of names on this slide, which I all have to thank for the involvement within this project, and everyone had a part in the work. So a quick overview. So the BARn project involved the three partners I mentioned previously, the University of Minnesota with Gary and Alison, then Ronja and Niels at the IPK in Germany, and a lot of people here at the University of Dundee and the James Hutton Institute in the UK. We've been growing barley in the fields, and you can see one of those nice plots over here in all three locations in 2 field trials in 2019 and 2020. So what are we actually looking at, and what do we want to identify? So the BARn project came from the idea of growing 200 two-row spring cultivars, and they've been selected based on the year they've been registered to the national list, which is a list advising on the best barley variety at that time. So we've got a wide range of barley cultivars from 1830 till 2016. And from these around 200 lines, we collected phenotypic data, expression data, and genotypic data. And the idea is to combine all of this to then identify genetic regulation of the barley yield. So I'm going to talk about each of these three subset phenotype data, expression data, and genotypic data, and then just briefly touch on the last bit because it's been taking a while to collect such an amount of data that we're only just now getting to the real data mining down. So phenotype. So this is a representation of a small tiny barley seedling just germinated in the field. It produces more and more leaves, and this is the first phenotype we scored, which is the growth habit. And you can see on the left side one extreme, which is very straight up plants with the leaves all pointing upwards. And you can see on the right-hand side, there are some varieties which show a very loose phenotype where the leaves are all falling on the ground and it's very white. So that was the first phenotype we scored in the field. Then the barley plant undergoes stem elongation and produces a flag leaf. And then the next time point, and then the next score is time point, and we looked when 50% of the plants in the field showed awn tipping, so the first awns appearing. The next step is then heading, when the whole spike emerged out of it. And then this is being elongated, so this put out of and the last step is then we scored senescence, so when the plant was fully mature. This also led to derived phenotypic measurements, like the time points from tipping to heading, heading to senescence, and tipping to senescence. Before we harvested the plants in the field, we then went to the field and did height measurements. So we measured the bottom of the peduncle, where the flag leaf started, the bottom of the spike, top of the spike, and top of awn. And again, we also did derived measurements, as you can see here. So total peduncle length, how much the spike actually emerged out of the flag leaf, the spike length and on length. And also we did spike culm ratio. Then the plants were harvested in the field and we collected 15 individual spikes per plot and did individual measurements on those again. So the first one was the total spike length again for 15. And then we also scored the number of rachis nodes. So each position where a grain is attached to the spike is a rachis node. And we counted all of them, and based on this we determined spike density for this. We also looked at infertility in this spike, so combined the infertile florets at the base and top for a score we called infertile floret edges, and the ones in the middle, and also then total infertile florets, fertile florets, and the percentage. The last bit was then taking the grains and putting them on the marvin and calculating grain length, grain width, and total grain area, and also 1000 kernel weight and kernel roundness. So in total we have 30 phenotypes collected across 2 years across 3 sites. Plus there was one polytunnel experiment done here as well. So there are a few measurements for the polytunnels. Next bit is the expression data set, sorry. So for this, there were 6 tissues chosen on, and each location did 2 tissues. So here at the University of Dundee and JHI, we did the peduncle and developing inflorescence. Ronja at IPK did developing florets at the green anther stage and 5 days post-anthesis. And Allison in Minnesota did seedlings which were grown on a plate and then harvested after 1 week, the top, the crown part, and the root part. And this was done for all 200 lines, and then we did Illumina sequencing and then the quantification against BART version 2, which is the new BARley reference transcriptome done by Max and Franchwan and Wenbin and a few others. The last bit is the genotype data we collected, and for this we used a variety of data sets. So first of all, we already had the 50K SNP data available. We combined this with whole genome shotgun sequencing, which was done by Ronja at the IPK. And we also had obviously the RNA-seq derived variants where we could, the RNA-seq data where we could then do variant calling, and we integrated all 3 data sets together. This also allowed us to do quality control, which was really important because we could remove outlier samples or switch samples. So what you see here in this plot is the correlation of the SNPs from one of the data sets, so the RNA-seq peduncle SNP data set, and this been correlated against the 50K SNP chip. And on the y-axis you see the percentage of correlation, and what you want to is all 200 lines to be up here, 95 till 100% best, that they correlate really nicely. As you can see, that's obviously not the case. And you've got a few samples with low correlation below 50%. So these are either sample swaps or wrong genotypes. So for example, down here at the bottom, the Invictus and Balga samples we know were swapped. So when we renamed them and then looked at the correlation again, they turned up to be around the 96% up there. So these are these are easy to solve, but some of them were just wrong genotypes, either in the 50K SNP or in the different tissues. The other thing we looked at was also heterozygosity, which you can see on the x-axis on the bottom in percentage. And you can see these ones, they show a lower correlation, but this lower correlation is due to this heterogeneity. And we suspect this is either due to combined of different samples or sequencing issues. So what happened is that each of these different tissues for the expression data set were combined of at least 3 different plants. So either there could have been already a problem with the seeds at the start that they've been swapped, or during the sampling process. And this would then result in variant calls which could not be clearly distinguished. So we could already clean up the data set so these wouldn't cause issues later in the expression data set as well, which was really good. So if after combining all of these data sets and removing our outliers for each of the 6 tissues, we ended up with over 32 million SNPs. We did a quite stringent filtering on those SNPs and then followed by imputation and tagging based on the linkage groups, and then pruned those down based on linkage again, so we wouldn't have a massive amount of SNPs which just contain the same information. And this led us down to 474,000 SNPs, which we worked on for the remaining bit. So the analysis, the main one we wanted to start with, consists of 3 different parts. So the first one is genome-wide association study. And this looks at the sequence association between sequence variation with the variation in the phenotype. So you can see here in this nice diagram, we have the genotypic variation, and we want to correlate this with the different phenotypes where we had these 30 measurements from. The TVAS is an association between the gene expression and the phenotype. So we want to associate how the gene expression is correlating with the phenotype. And the last bit is the EQTL, which looks at the genetic variation and correlates this with the gene expression variation, or looks at associations between those. For GWAS, we used GEPIT with 2 different modules, BLINK and the mixed linear model. For TWAS, we did a linear model, and plus we included covariates to account for variation in the expression data set. And for EQTL, there's a program called Matrix EQTL. And again, we included covariates in this part for the expression. This sounds quite, dry. So I thought I'll give a more detailed overview of what it actually looks like. So this is what the analysis for each of these parts for one individual example would look like. So the one on the left is the EQTL analysis. And we're looking here at one specific SNP on chromosome 2H at this position. And there are 2 alleles in this population, the C allele and the T allele. And on the y-axis, you've got the expression for one of the candidate genes. And you can see that the C allele is associated with lower expression and the T allele is associated with higher expression. And you could do an individual t-test for this, and this is the p-value for it, so it looks very significant, highly significant. The next bit is the GWAS. Again, you've got the same SNP as in the EQTL at the bottom with the C and the T allele. And on the y-axis, you've got the grain width as one of the 30 phenotypes in millimeters. So you've got variation between 3.4 to 4.2. And again, the C allele seems to be associated more with a smaller grain, and the T allele more with a wider. And again, you can do an individual t-test. This is the p-value. The last bit is the TVAS, where we have the grain width across the x-axis on the bottom, and then the expression of the gene from on the y-axis. And again, there's in this case there's only a slight trend of which you can see. So it seems that lower expression is correlated more closely with a smaller grain, while higher gene expression with a wider grain. So this is just one example of one SNP, one gene, phenotype, and one gene. But we have around 20,000 expressed genes in each of these 6 tissues. We've got these 30 phenotype measurements, and we've got over 474,000 SNPs. So you can imagine that's quite a lot of genes and different GWAS, EQTL, TWAS numbers to look at. And this is where we just started. So I'm just going to give an overview about some of the results. A first kind of teaser. So for the EQTL, what we are actually interested is in identifying the regulatory mechanisms underlying this gene expression. So if you have a SNP, a genetic variation within the gene or in the promoter region that impacts on the gene expression, we're calling this cis-EQTL, as we are suspecting that this gene is then causing these different gene expressions. if the if the SNP is somewhere else on the genome, further away on the chromosome or on a completely different chromosome, then this is called trans-EQTL. And this is most likely due to a SNP in a transcription factor, for example, which is then impacting the expression on the gene of interest. So we got quite a high number of significant EQTL, between 8.5 to 9 million. If you go down to the significant SNPs, you can see for the cis they're between 2. They're around 2 million for each of these two tissues, but each gene has potentially multiple SNPs within the promoter region or within the gene. So this narrows it down quite dramatically then to 5,000 genes, which are of interest from now on. And we want to have a closer look at. For the trans-EQTL, there are even more significant SNPs, but you can see that the number of significant genes is a lot lower, which makes sense. So we're expecting more genes under cis regulation than under trans regulation in general. So this gives us a big group of genes of interest. And I'm just going to show you the combination with the TVAS results here, or one example with the TVAS result. So this is a transcriptome-wide association study where we look at the association of gene expression and the phenotype. And this is an example of the grain width. So what you get as a result is a Manhattan plot you've seen previously with the GWAS results. And this has 88 genes above the significance threshold over here. And you can see, for example, there's a big cluster of genes on the chromosome of 5H. So the question we're now wondering is, are any of these genes we've just identified for the sativas under cis control in any of our tissues? And the answer is yes, there are 78 of those genes are, for example, under cis control. And how can we further narrow this down, find interesting patterns? What can we further identify with that? And for this, one of the examples which we'll follow on, one of the analysis we'll keep doing as well, is a network analysis on the gene expression. And there, for example, 33 genes of those 78 are in one module. And I did a quick network expression analysis, and this is one of these graphs you get then. And you can see I've colored them based on the chromosome they're coming from. So the light blue ones are the genes on 5H actually. And there's a very tight cluster here in the middle, which seems to be the main genes involved. So that's one of the examples on how we can go further and identify genes of interest. There are also obviously other options. So we can do a network analysis with the remaining 5 tissues, also combine the TVAS of all 6 tissues, see which genes are actually correlated with the expression in all of them, and so on. So there's quite a lot to do, and this is just one of the examples on how we started. So there are other stories, as I just said. We'll want to do a network analysis across all 6 tissues and hopefully identify with this also tissue-specific genes and then genes consistently expressed across all tissues. I didn't even touch on the genome-wide association study, which we definitely have, where we don't have results for all of the phenotypes, so that we don't get significant associations for all of them. But for quite a few, we get some nice peaks. And we definitely need to follow those up as well. And then this is one of the examples. We had just said previously, the 5H cluster looks of interest. And I've done a cluster analysis only on the variants on 5H. And these split across the year they've been registered to the national list. So the one on the left-hand side are the years before 1990, probably around this. And these are all the new varieties. So there's a clear split of SNPs based on year registration. And this can also be seen in the gene expression, and this fits with the cluster on 5H. And this is one of the things we're definitely going to follow up. Starting again. So I'm going back to my start screen because that's me now giving you a quick introduction into the Big Barn project. And I've got to thank a lot of people, so there've been lots of work done here at the Institute before I even started the project of different people. And obviously Ronja and Alison, who've been part of this as postdocs from the beginning and introduced me to the whole thing. As you can see, we're just getting started on the deep analysis, and it'll be exciting to see what we get out of it next. But that's me for now, and with this, thank you for listening.
Thank you very much, Miriam. That was absolutely fascinating. So I'm sure that there's going to be a lot of questions once everyone's finished with their many applauses that are coming through. So who would like to start? I'm sure everyone's. Don't be shy. So I think whilst people are kind of digesting all the information and whatnot, so there were quite a few things that I wanted to ask actually throughout your talk, and then you answered them as you went on. So I suppose you'd mentioned about combining all the different tissues. So all this data has also been combined between the 3 different sites as well. Is that right?
Excuse me, what's been combined?
Sorry. So sorry. So just for me to clarify, so all of the data that you're looking at, this is from the 3 different sites that are all being combined across all of these different cultivars. Is that right?
Yes. So we've got the seeds were sent from here to IPK and Minnesota. So we're all working on the same seed stock in theory, and we've all done our individual parts, but then combined each of these. So the genotypic data is, for example, completely combined. The phenotypic data at the moment we are working on BLUBS, which stands for biased. I wrote it down somewhere. Unbiased. Oh gosh, I need to look it up. Sorry. So they've been combined. But we'll look also into the individual ones because there've been differences in Minnesota to comparison to us here, especially. They've got a different kind of season.
Fantastic.
Yeah.
And have you noticed any mild variations between the different sites then? So although they're supposed to be or identifying as the same cultivars, do you notice kind of regional differences between them?
Yes. So
Yes.
I've not plotted the phenotypic data, which is one of the next step I'll go back to and do. But especially Minnesota was earlier, so they took a bit longer at the beginning, but then they kind of shoot up because they get the warmer weather faster than we get in Germany or here at Dundee. Especially the Dundee times were the slowest ones, which isn't unexpected. So you can see, especially in the time points differences, but also in some of the other things. Plus there was the infertile one from 2019 we had to take out from the Minnesota group as well, because they had a bit of weird weather, so the spike didn't emerge fully, so they were kind of screwed a bit.
That's not ideal. We've got a question from Ruth. Ruth, do you want to read your question, or would you like me to read that out? So Ruth is asking, do you see a clear difference in any of the phenotypes between the pre or post 1990 varieties?
Big difference, which is obvious, is the actual size of the plant. The other ones are more subtle, I think, the differences, but especially with plant height, and if you look at the oldest varieties in the field, we had to be careful with lodging a bit, while the newest ones obviously are not that much of an issue. So that was one of the biggest obvious phenotypes. Everything else is, yeah, more subtle. I need to have another look as well in the different sites, which I wanted to have another look at as well, which will be interesting to see if there are bigger differences in Minnesota than here.
I think we might have a question from Claire as well.
Yeah, or more a comment and a kind of a question, I suppose. Miriam, it's just to remind you, particularly in light of what you've just said about lodging, that, you know, we have an RNA-seq data set from stems that will complement what you have. Many of the same cultivars will be in there, I imagine. So it would certainly be worth comparing data at some point, and particularly for things like lodging or height, which I think we also measured in our analysis. But what you have there sounds really beautiful, really nice resource.
Thank you. Yeah, I think I actually might still have access to the data somewhere.
Well, we'll have done some analysis, so we can share with you what we've done, but you probably do have access to it, yeah.
Yeah, it'd be interesting.
Thanks, Claire. And does anybody else have any questions or comments at all for Miriam? Okay, I don't see any hands up. Miriam, I think you're off the hook.
Thank you for giving me the chance. That was good.
Oh, I think we actually do just have one just now. So from Reza.
Yeah, hi Mariam. It's a very nice talk. Just question actually, it's an enormous amount of data you are going to generate. It's 30 different phenotypes and expression data. And how are you going actually to combine this? Because the current actually the analysis is going to take only one set of data and combine it. How are you going to combine all 30 phenotypes and the expression data? Is it any plan for that, or it's going to be done?
We will put up a lot of this onto Germinate, I think, as a database to distribute with people. So I've spoken with Sebastian about this, but I've not added it to Germinate yet. But this is the perfect place to add our phenotypic data and genotypic data. With the expression data set, we'll have to see how we'll make that available. It'll all be uploaded anyway to the ENA, for example, but I'm not quite sure how we'll make that one available yet. But yeah, it's quite a lot of data which we'll work our way through. At least we have 3 people to have a look at all of that.
I got another question. Is it okay to ask? Is it how do you incorporate the environmental data so they can get at the end actually GYE and the effect of a genotype, actually environment?
Yeah, we need to have another look at this. So we've not directly included it. It's part of the module selection, and I have to say the GWAS bit is the one I'm less confident on. That's been mostly done by Ronja and Alison. But we have been looking at this, and at the moment we're kind of accounting it by using these 3 different locations as well, because yeah, that's taking it kind of out.
Thank you.
Thanks, Reza. Okay. And any other questions or comments for Miriam today?
Just drop me an email.
Yeah, I think that's a really good idea. Well, first of all, thank you so much for such a really fascinating talk, and also for keeping to time and for explaining things in a very accessible way. Thank you. If we could all come together just to thank Miriam for her time today. Okay, thanks very much. So we'll see everybody again in 2 weeks' time for the next DPS seminar. And thank you again, Miriam.
Thanks.
Take care, everybody.